Assalamu alaikum. You are watching Microbiology by Nouman. In this video, we'll talk about Staphylococci and classification of Staphylococci into Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus. We will also differentiate between these three species of Staphylococci. So let's start. So Staphylococci are round-shaped bacteria, and round-shaped bacteria in microbiology we call them as cocci. When we see staphylococci under microscope, they have a arrangement called grape-like cluster, right? Irregular arrangement, just like the cluster of grapes. Staphylococci don't have a proper arrangement, so their arrangement is called irregular grape-like clusters. Also, staphylococci are gram-positive cocci. That means after performing gram staining technique, a gram staining is a very important staining technique in microbiology. After gram staining technique, staphylococci appear in the form of purple color bacteria that are round in shape. So they are purple in color and round in shape and have grape-like cluster symmetry or irregular symmetry. Also, staphylococci are facultative anaerobes. That means they can live in the presence of oxygen and in the absence of oxygen as well. They are non-motile and non-spore forming. Another important characteristic of staphylococci are they are catalase positive. Catalase is an enzyme that can convert hydrogen peroxide into oxygen and water. So catalase is one of the virulence factor of staphylococci. That means catalase play a role in causing disease in humans. Just remember that catalase is an enzyme that differentiates staphylococci from streptococci and enterococci. Streptococci both of these are gram-positive bacteria, but they do not contain this enzyme. They are catalase negative. Staphylococci are further classified or categorized into groups on the basis of enzyme known as coagulase or coagulase. Coagulase enzyme can convert fibrinogen into fibrin. And resulting in a clot formation. Coagulase is also one of the virulence factor of staphylococci and play a role in causing disease in humans or in animals. On the basis of this coagulase, staphylococci are categorized into two groups, into two categories. Coagulase positive staphylococci includes Staphylococcus aureus, whereas those bacteria, those staphylococci that do not contain this coagulase enzyme, they are known as cons, C-O-N-S. Coagulase negative Staphylococci. Coagulase negative Staphylococci includes Staphylococcus epidermidis, and Staphylococcus saprophyticus. Due to space shortage, I am writing like this, but they are written in straight form. Staphylococcus aureus. Talking about Staphylococcus aureus, it's a pathogenic bacteria. That means it causes diseases in humans. When Staphylococci grow on surface of culture media on inside lab, culture media is used for growing bacteria. When Staphylococci grow on culture media, they produces golden colored colonies. Golden colored colonies due to staphylocanthin. Also, Staphylococci produces beta hemolysis on blood agar plate. That means Staphylococcus aureus, when it is growing on blood agar plate, it can completely break down red blood cells from blood agar plate. So, such a hemolysis is known as beta hemolysis, complete hemolysis. So, talking about Coagulase negative Staphylococci, first we'll talk about Staphylococcus epidermidis. It's a normal flora of human body. Staphylococcus epidermidis is a normal flora of human body. That means it's a good bacteria that protect our body against other bacteria. So it's a normal flora of human body, but certain strains of Staphylococcus epidermidis can cause infection in humans. Only those strains that contain glycocalyx, only those strains of Staphylococcus epidermidis containing glycocalyx can cause prosthetic valve endocarditis or prosthetic infections in other body parts, but only strain that containing glycocalyx infection causes infection in person having prosthetics in their bodies. Staphylococcus saprophyticus is only causes infections like urinary tract infection. To differentiate between Staphylococcus epidermidis and Staphylococcus saprophyticus, we can perform a test known as novobicin. Novobicin is a drug that is used to differentiate between these two bacteria. Staphylococcus epidermidis is sensitive to this drug. That means if this drug is present, Staphylococcus epidermidis cannot grow. Whereas Staph saprophyticus is resistant to this drug. In the presence of this drug, Staph saprophyticus can grow. Just keep in mind that Staph epidermidis produces white colored colonies. Staph aureus is also novobicin sensitive. Also, Staph aureus can ferment mannitol, whereas Staph epidermidis and Staph saprophyticus cannot ferment mannitol, right? So they are non-fermenter of mannitol. Mannitol is a sugar. Staph aureus is also DNase positive. whereas Staph epidermidis and Staph saprophyticus are DNase negative. These bacteria, these Staph epidermidis and Staph saprophyticus do not contain this DNase enzyme. So if we briefly describe Staphylococci, they are gram-positive cocci. They appear in the form of irregular grape-like cluster, purple in color and round in shape after gram staining. They are non-motile, non-spore forming, facultative anaerobes, Catalase positive, and on the basis of Coagulase, we categorize Staphylococci into two groups, Staphylococcus aureus and Coagulase negative Staphylococci, which include Staphylococcus epidermidis and Staphylococcus saprophyticus. Staph aureus is a gram-positive cocci, catalase positive, coagulase positive, pathogenic bacteria, produces golden color colonies on agar, causes beta hemolysis on blood agar plate, is novobicin sensitive, mannitol fermenter, and DNase positive. Staph epidermidis is a gram-positive cocci. It's a catalase positive and coagulase negative. It's a normal flora of human body. Only stains of Staph epidermidis containing glycocalyx causes infection in people having prosthetic in their body and cause infections like prosthetic valve endocarditis or prosthetic infections of other body parts. They are novobicin sensitive, produces white color colonies. They are DNA is negative and non-mannitol fermenters. That means they cannot ferment mannitol. Staph saprophyticus is gram-positive cocci, catalase positive, coagulase negative, causes urinary tract infections. They are novobicin resistant, cannot ferment mannitol, and DNA is negative. That's it from this video. If you have any question, you can ask in the comment section. For more microbiology content, please like, share, and subscribe my channel. Thank you.